things that Kristen referred to and Ernst referred to a little bit. This substance called colored dissolved organic matter. I'm going to talk to you about that and cover a few of the applications of studying this material in different environments. And mostly we use fluorescence spectroscopy to do it because it's really, really sensitive. Anybody know where this picture is? Ernst does. I just told Ernst, so he doesn't count. He didn't know what it was. I had to tell him. This is the mouth of the Mississippi River. And you can see this brown stuff here is CDOM, brown stuff. We have a lot of it around here. So I'm going to talk about what it is, why it's important, a little bit about how we measure it, but not really many details, and then just give you a few examples from various environments that we've studied over the years. So what is it? It's colored dissolved organic matter, colored CDOM. CDOM, colored DOM. Some people get fancy and call it chromophoric because that just means it bears color. Older names are Gilvan. My favorite is Gelbstoff, which means yellow substance or yellow stuff. But basically it's thought to be composed mostly of humic, fulvic, and hydrophilic acids if you go back to some of the more complicated chemistry. Why is it important? Because it's yellow. I know, it's kind of a no-brainer, but that is why it's important. This happens to be, is this your instrument, Chunmin? In the Mississippi River? Were you on that cruise in the Mississippi River? No. So the penetration depth here for the optical sensors was, I think, 1.5 inches. Yeah. It's important because it absorbs blue light. Okay, that's what phytoplankton need to grow, and you can see how much of it is there. The phytoplankton are not getting a lot of light in this environment. It's strongly fluorescent, and fluorescence is a very sensitive technique, which means that we don't have to concentrate our samples, and we can measure it in situ without any sample processing. It's also highly reactive in sunlight. I didn't really give too many examples of why that's important. Sunlight breaks it down, but the sunlight also helps mediate trace metal interactions, making the trace metals more or less available, and participates in redox reactions of trace metals. So that's one of its important things. This slide was put together by one of my former students, Robin Conmee. And so the ecological importance, I kind of said, it's important in controlling light penetration. It interferes with satellite algorithms for chlorophyll because it absorbs blue light. But it also provides UV shading in environments where there isn't a lot of CDOM. So this is important in coral reef environments and some of the blue water environments. And really just a small change in CDOM can have a huge change in terms of the UV shading effects. It's important in carbon cycling because you can trace this as you can trace it as a water mixer. It is a component of the carbon cycle, and in some environments it's a nice proxy, especially freshwater systems. DOC concentration and CDOM concentrations are correlated at 99% correlation coefficient. So in some environments it's a very easy way to look at the amount of carbon and where that carbon's going. I alluded to the fact that it's important in metal binding, and so in some environments, if you can trace the CDOM, you may also trace any pollutants that are coming in with that freshwater source in the CDOM. Deep ocean, just in the last 5 years, I think, it's been shown that high CDOM concentrations in the deep ocean are totally correlated with apparent oxygen utilization, the implication being that in the subsurface ocean, microbial activity is producing this organic matter, and it's highly stable over long periods of time. But it can also be an organic nutrient source. I said that it interacts strongly with sunlight, so if the sunlight starts breaking down the organic matter, it makes smaller molecules that then the microbes can take up. So societal importance, it's used as an indicator of ecosystem health. In the estuaries locally, especially because, Ernst talked a little bit about eutrophication, but in our estuaries they're often so brown that there isn't much phytoplankton growth. It's really the CDOM that is limiting light, and as they use seagrass as an indicator of ecosystem or estuary health. If there's a lot of CDOM, there's not any light getting down to the seagrass. And the CDOM is coming in from completely natural sources. It's not a man-made substance. It's coming in from soils in the marshes and the swamps that we have. Since I started doing this work, there's been a lot of work on drinking water quality that has a very, very huge application in drinking water quality, and partly because the organic matter actually will interfere with the chlorine, and that makes a carcinogenic thing. So if there's a lot of organic matter, they can't chlorinate the water. They have to shut down their intakes. It's very sensitive to wastewater contamination. There are a couple of amino acid-like signals that come in with wastewater. And this is having lots and lots of applications in places like Australia and probably the US Southwest, where they're working on reclaimed water systems for drinking water. Very sensitive. You can monitor the process in line. It correlates with salinity, so you can use it for estuary mixing models. A couple of our students here have used it to test some of their models in Tampa Bay. And it's a water quality parameter that you can measure from space. I think Chunmin is doing some of this. You can measure it at 412 and 443, and of course the new satellites are going to go down to 355. So I'm sorry I forgot to put a spectrum, but basically the absorbance spectrum of this stuff looks like this. So it's really, really high in the UV, and there are not really very many features in it. And you can use it for water source tracking in the open ocean, pretty much anywhere. I challenge you to give me a sample that I can't measure CDOM in. Anything that you can think of, it's pretty much in there. So this is just kind of a schematic of where it comes in. Mostly it comes in from rivers. It's humics that are in soils and some pollutants. Productivity produces some distinctive signatures. Sediments are high in humics and amino acids. Upwelling brings deeper water, and the deeper water is high in humics because it's not all bleached out from the sunlight. The main sink of it is sunlight, and the major product of photo bleaching is carbon dioxide. So it can be a source of CO2 in coastal areas, just the photo bleaching part of it. Photobleaching or photooxidation does produce these low molecular weight carboxylic acids, which then make the microbes happy, and then they produce other CDOM that's a little bit different in character. Some of the other places where it's been measured are rainwater, hydrocarbons are fluorescent, sewage, agricultural waste, lignin, mangrove leaves, seagrasses, vitamins are fluorescent, and there's some evolving applications in groundwater. This is a satellite compilation from Dave Siegel, published over 10 years ago now, of CDOM distributions globally. And this is measured using probably the 443. Sorry, I didn't put that on there, but it's measured using the satellite response return at 443 nanometers. And the top one, this is winter and this is summer, and this is his CDOM concentration in an absorption coefficient per meter. And the thing to notice here is that the blue areas here are where the oligotrophic areas where the material is all bleached out. So it's warm surface water with thick mixed layers. And you see that moving towards the poles in either direction, you get much higher concentrations. The old CZCS imagery of chlorophyll was really measuring CDOM in the polar regions. So it was something they couldn't correct for at that time. And now we're able to correct for it. And this bottom one is the same winter and summer, but now this is the percent of CDOM that makes up the total signal of 443. And what you see here, the numbers here, this is like 50% is this pale peachy color, and that's 100%. And so what this is showing is that almost all the signal that's coming back from the ocean is actually from CDOM and not from chlorophyll. So that was really important. It took how many years for the biologists to accept that? Do they accept it yet? It was a pretty big thing. Nobody really wanted to believe how pervasive it was. So what is absorption and fluorescence? I stole this slide from my student Anna, who just defended last fall. I couldn't find mine, and hers were much prettier anyway. So the process of absorption is when a molecule absorbs energy, and in this case goes to a higher energetic state. And what happens after that is usually relaxation to a lower energetic state. And when the molecule comes from the first excited state back down to the ground state, it's called fluorescence. So the molecule absorbs a quantum of light, it relaxes a little bit, then it releases a quantum of light at a longer wavelength at a lower energy. And that's what the process of fluorescence is all about. Not all molecules fluoresce. Oops. I think I took out the last bullet. Not all molecules fluoresce. So this is an example of what our results look like. We call it an EM, an excitation emission matrix. This is quinine sulfate, which is the standard that we use. This is the excitation wavelength here. So what we do is we put in a sample of water, and it's just filtered water. That's all you do. Filter it, put it in your cuvette. Isn't that easy for those of you who don't like wet chemistry or, like spending a week in the lab preparing your sample? With this one, you just put your water in there. And you don't have to filter it, but we usually do. So we put it in our cuvette, and we shine a wavelength of light on it. And then we scan to get all the wavelengths, all the light that's coming out at different wavelengths. And that's what this axis is, the emission. So this is a short wavelength here, like 250, and then the emission peak looks like this. And we just compile these. We have anywhere between 40 and 50 scans, and we get this matrix. This is a 3D plot. We usually look at them in contour, like a map. So the parameters that we look at are the fluorescence intensity, the height of this peak. You can look at the position of the emission maximum if you fix an excitation and then scan the emission, but you can also look at the position of the excitation maximum if you put your monochromator here at 450, the emission max, and then you can scan across the excitation. So you can do it either way. The new instrument we have in the lab now is actually a CCD array, so it just goes like that, takes a snapshot of it, so it's very nice. And these are just kind of some of the areas, the peak position and the intensity that's allowed us to discriminate water types and contaminants. These are just some of the areas where we find peaks. These short wavelength peaks are related to the long ones, so that they're actually pairs of emissions for a particular component. Most compounds have a very short excitation or absorption maximum down below 260, but they also will have a longer one out here. And so they have dual excitation max, but the emission occurs at the same time, and that's just physics. So these peaks here tend to be in rivers. Sometimes it's even longer. The M peak here was first found in seawater, but now they're finding it in agricultural waste, and it seems to be associated with recent metabolic activity. The B and T peaks are they're kind of amino acid-like. B is like tyrosine and T is like tryptophan, and they come in and out depending on what's going on in the environment, but they're also indicators of sewage and waste products. So overall, in looking from across this slide was actually developed from sites all over the globe. This was one of the first ones that I did. And I apologize, the symbols are really small. But let me go back one. So you basically have a couple of like a family of fluorophores that falls along this axis. We've identified M and C because we can see them in samples as pure, but they actually, in some of the really fresh rivers, you'll get a maximum almost out here. And now when people are using mathematical techniques to separate out the peaks that are in here, we see like a family of things. So if we look at the position, my early work was just looking at the position of the maximum in this region here, and just taking the excitation maximum and locating it in excitation and emission space. And that's what this plot is for samples from all over the world, the Columbia River, Hawaii, Amazon. And what I found was that there was a gradation from the terrestrial being at emission 460 and excitation around 350 or 60. These are pore waters and river samples. These are estuarine samples, and these are marine samples, especially ones that were taken where there was high productivity. So we could discriminate the kind of environment that the water sample came from just from doing this little simple scan. It now takes 5 minutes to do these. It used to take me 2 hours. And even on a smaller scale, this was a student, Jen Bamey, who did this for various local rivers, and you can see this is now showing the short wavelength and the long wavelength maximum, and you can see that, that the rivers separate out nicely from some of the bays and the Gulf of Mexico. So it's a really nice technique for separating water sample origin. This is kind of some extremes that I found from the Amazon River, fresh riverine. It's nice and fat and happy. This is a partially bleached sample. I call it the carcass because you see that it's just like carved out here. It looks like the turkey after you've served it up for Thanksgiving. And this one is a really very bleached one, really low. Now this is down like the maximum is 1, this one's 4, and this one's, well, these aren't real numbers, but it's probably like, rivers can get as high as 600 for that peak. So this one's from an oligotrophic area, and you see what happens to the contours. Not only are they moving from here to over here, but they're also now not very round, and they're all bleached out. So you can also tell something about the history of the water mass based on this little picture. In terms of the intensity, fluorescence intensity in different environments, these are just kind of relative. Rivers, coastal marine, oligotrophic marine, deep waters actually are higher, and pore waters can be really high. So just kind of as an indicator, you can see that this, this transition here would give you a nice mixing line, and in fact, that's what happens. This is now the intensity of that peak C and salinity. Now this is just salinity 30 to 40. So there's a really strong relationship down to 0 in most coastal areas. But you can see for our study here on the West Florida shelf, the Mississippi River is distinctive, has a much lower slope. The end member concentration is pretty constant from year to year. We've measured that. The West Florida shelf rivers are higher. Not sure, I think this is the Shark River over here. And a couple years ago, there was that black water incident that people were, the fishermen were all upset because they thought there was oil in the water. And we were able to use our background concentration to say, well, this black water really looks kind of like the Shark River. It's not really falling off of our mixing line, so we're not too concerned. This is kind of interesting here. There was a time, one particular cruise or 2 cruises, where we could see Mississippi River water coming into the area off Charlotte Harbor. And then this is also an interesting thing where now we get a positive relationship. Florida Bay shows this in the Charlotte Harbor region, and we published a paper on this a couple years ago, where it looks like there's new production of organic matter at the high salinities associated with, we think, water coming off of the tide marsh banks, tidal banks. And this is that same plot before that I showed you, and now this is that satellite picture of the of chlorophyll as an indicator. The red part, that's showing you the area there in Florida Bay. This is showing you the area off the Shark River, and this little filament here was shown to be Mississippi River water. So it's kind of a nice way of kind of figuring out what's where your water is coming from. My student Robin Kami tried to use this in groundwater studies here in the Tampa Bay area. And this is a little bit complicated, but not terribly. The blue line is the mixing line for the Manatee River. This red line is the Hillsborough and Alafaya rivers. And this is the mixture that you get in Tampa Bay. And she was looking at different levels in the Floridan aquifer under the Tampa Bay. She's working with Swift Mud on this. And you see that these points here are from the surface aquifer. Well, that makes kind of sense. They look kind of like the rivers because it's just the fresh stuff that's run off in the surface. But in the deeper aquifer, I think it's, yeah, oh, good. So these are samples taken at less than 150 feet. These are the ones greater than 150 feet. And what happens within the aquifer is that the organic matter gets remineralized, that regenerates the nitrate, but the organic matter goes away, and so the CDOM concentration falls because that's what it is. So she used that. It was a really nice study. Anna tried this in the Crystal River, and it was a lot more complex because the aquifer wasn't, it was an unconfined, unconstrained, what's the right word? It was undifferentiated. So what she found was that there was a really important influence of seawater coming in offshore and kind of mixing with the aquifer water on one side that was giving high CDOM because the seawater was high there because of the rivers. And on the other side, the CDOM was low, but it had high inorganic nitrogen and some indicators of sewage pollution. And so what was happening was that the seawater was mixing with the aquifer and coming into Kings Bay, which is north of here where all the manatees are. But Kings Bay is spring-fed, and so the whole salinity relationship was opposite. There was the low salinity, the low salinity had low CDOM because it was coming from the groundwater. And so it was a whole mixed-up system, and I couldn't find one slide from her thesis that would really let me explain it. So, but if you're interested, that was a pretty nice study that she did. This is a picture of a thin section of coral. And many years ago it was shown that the coral had fluorescent banding in it. And this has been related to river discharge. And so looking at a fluorescence microgram of thin sections of corals, you can get a historical record of annual river discharge. This is from a book chapter that's coming out soon. So lately we've been working with the oil spill response as well. This is a slide that Robin put together for one of our earlier presentations at an optics meeting. Fluorescence has been used in oil spills for more than 30 years. The Coast Guard has for a long time just been running fluorometer from their aircraft, doing aircraft surveys, and because it's so sensitive, they can get a quick idea if there's some kind of oil spill in their area. You can detect it on the surface, you can detect it in the water column, and you can detect it in the sediments using real-time fluorescence instruments in various configurations. And the real-time measurement is critical for decision making. This just shows some of the instruments, you know, benchtop, UV vis. Of course, they're more real, true chemical analyses for this, but they require some preparation. They're portable, submersible. The list is a particle sensor. These have all been used in oil spill response. Here's a diagram of how the Coast Guard used to use it with airborne scanners. And then this is a picture. This is probably Chun Min's picture. I don't know. Yeah, of what this looks like from a satellite. I think that's a reflecting true color. Yeah. So it's very sensitive, it's very fast, and it's very accurate, which we've just demonstrated in a paper coming out soon in Marine Pollution Bulletin, I think, where we did some wave tank studies and we calibrated different fluorometers and did the chemical analysis. So we've demonstrated that the fluorometers are very sensitive. And this was mostly looking at the buried stuff. So this is, again, that same true color image. And these are our EAMs now from, you know, our fluorescence scans. This is the excitation wavelength. This is the emission wavelength. And what you see is this characteristic, like bullseye kind of spectrum here. It's not, CDOM would be spread out kind of along an axis like here. And this is from oil, and they're very high concentrations. This is like 700 now. The white spots are where we cut out. The technique is so sensitive that the dominant signal in these samples is scattering from water, the Rayleigh and the Raman scattering, and that messes things up and makes it really hard to look at. So we just kind of cut that out and look at what's underneath. So definitely oil there. There was a lot of interest in the subsurface oil plume. People were doing profiles, so I'll start up here with this profile. Let's see, the green is temperature. This is salinity. The blue is oxygen, and you see this little subsurface blip here. And this is from one of the CDOM. This is a wet lab CDOM fluorometer. And so people were observing this little blip here in oxygen and fluorescence at somewhere between 1,100 and 1,300 meters. It turns out it was on a density surface that was plus or minus 0.1 sigma T unit. So it was a very defined density surface. The depth varied a little bit, but was always on that surface. And now these are EAMs that we took. This is 3.4 meters, so this is surface 1. This one is kind of from in here, kind of in between. This looks like, normal CDOM. This is the one that came from 1,150 meters. And again, you see this telltale characteristic, really short emission maximum around 320 to 340, indicative of oil being in that spot. So this is something you can now do in profiling mode, potentially. Some new sensors are being developed based on this work and all of that. So just kind of to reiterate some of the freshwater challenges that people are using CDOM fluorescence for, the water quality monitoring. I know I was told by the guys down at Peace River who were bringing in water from the Peace River to use as drinking water. When the absorption goes above a certain point, they shut off taking water from the river, and they just have the inline sensors. You can identify some pollutants like oil, but there's some other ones. I think I took that out. I'm not sure. You can also look at optical brighteners that they use in detergents. And so one of the students at Pam and I had together had developed a underway in situ method for that. We're looking at groundwater input and mixing, drinking water quality, land use change. There's a lot more organic matter coming off now that the permafrost is melting and eroding. So there have been studies in England and some of the other in Scandinavia showing that the CDOM is increasing with climate change, and that's influencing the light budgets in the local coastal waters, which is important, and freshwater budgets. But the nice thing is that these land-sea continuous observing networks, it's really easy to measure this routinely and quickly, and so it's really nice for that, and now it's being used in some models and budgets. So the book that we've been working on for about 4 or 5 years is actually finally coming out this year. It's the first complete compilation of everything from the physics of fluorescence to the chemistry of the materials in the fluorophores, to how to measure it, to applications, to microbial influences. So that's about to come out, and these are just 2 pictures of this is the Arabian Sea, and this is the mouth of the Shark River. The other application that I forgot to mention is that they're actually using it now to verify that tankers are exchanging their ballast water at sea. So it's very sensitive. They can monitor it on the ship. They can go and use it because when the ship's in port, it's taking on ballast water that's coastal, and they're supposed to exchange that ballast water in the open ocean so that they're not carrying any harmful organisms. So it's used for that. I forgot that. I took out a bunch of stuff so that it would fit in the time. It looks like I took out too much, but we're back on schedule. Just wanted to thank Robin and Anna because I swiped some of their slides. Thank you.
A couple of questions? Yeah, fine.
Can you detect the relative contribution of paper mill effluent?
Yes. I haven't looked at paper mills. They have a very distinct signature, though. So, sometimes a paper mill uses pigments or something like that to, like make pink tissue or something. So there has been work done in paper mill effluents. Yeah, absolutely.
More questions? Okay. Thank you again.